Good morning students. Today we will study about the antibody production. So antibodies, they are the protective proteins which can bind to a antigen. When we are dealing with the production of antibodies, at that time we need a antigen. In the presence of antigen only a antibody is produced. So when we are dealing with the production of antibodies, there are four different steps for which a antibody production has started. So in the first step it is the antigen presentation. When an antigen invade our body, at that time the macrophage they get activated and they take in the antigen by endocytosis. So when a macrophage is activated, it is taking the antigen by endocytosis. Then a macrophage processes this antigen and attach it to the membrane protein called the MHC protein. Now this MHC protein, it will move this particular antigen on the cell surface. It is displaying this particular antigen on its cell surface. So this is the first step in antibody production, that is antigen presentation. It is taken by a macrophage, then it is processed and then it is moved to the cell surface by the MHC protein. Then comes the next step, that is the activation of helper T cell. When we are dealing with the activation of helper T cell, this processed antigen by the macrophage, then it will activate a helper T cell. These helper T cell have receptors on their cell surface which can bind to the antigen which is presented by a macrophage. So when this activated T cell, it will bind to the macrophage, then at that time this macrophage, it will send signal to this particular helper T cell and it will activate this helper T cell. As the helper T cell it get activated, at that time this activated helper T cell, then it will activate a B cell or a B lymphocyte. So the B cell have antibodies in their cell surface membrane, means they are having a receptors, they are having a antibody structure on their cell surface. So when the B cell they are activated by a helper T cell, at that time they will bind to this particular helper T cell and this activated helper T cell then send signal to B cell to get activated, and as the signals are transformed to B cell, then B cell get activated. The helper T cells, as it is sending the signal to B cells, this B cell get activated, and when the B cell activated, the fourth step comes forward, that is the proliferation of the B cells or proliferation of the cells. At that time the activated B cell starts to divide by mitosis and they forms a number of cells which are similar, they are known as clones of plasma cells. When these plasma cells they are produced, at that time these plasma cells they are capable of producing a large amount of antibodies. So the number of plasma cells which are produced by the activation of B cell, they will start the production of antibodies. So now when the number of cells which have divided and they have given rise to a production of antibodies, there are number of cells which are also present which are not able to start production of antibodies. So some cells which stop dividing and secreting antibodies, then they are converted into a special type of cells which are known as memory cells. So these memory cells, they are capable of having a memory of a particular antigen which has invaded our body. So when the second time this particular antigen invade our body, at that time these, due to the presence of these memory cells, they give rise to a quick response, they give rise to a fast response in production of antibodies against that particular antigen. So large amount of memory cells, they will remain in our body for a long time. So now we come on the summary of the antibody production. For this we need a macrophage and a antigen. So as the antigen invade our body, the macrophage it will process this particular antigen. So as the macrophage it is activated, then it will make some changes in this antigen and then present it to a helper T cell. So the helper T cell it have receptor on its cell surface. So this helper T cell it will bind to this processed antigen by a macrophage, and then this processed antigen it is taken by the helper T cell to the B cell. Now due to the presence of this antigen on its cell surface, the B cell get activated, and when the B cell get activated, it start dividing and it produce a number of similar cells which are known as clones of plasma cells. So now the number of clones or number of plasma cells are being produced. Now they will give rise to the production of antibodies. So when the plasma cells, they have given rise to the production of antibodies, at the same time the number of cells which stop dividing and stop producing antibodies, they are converted into the special cells which are known as memory cells. So memory cells are the special cells which have the memory of this particular antigen which has invaded our body, and for the second time when this antigen is invading, at that time these memory cells, they will give rise to a quick response, and we get recovered from a particular disease or an antigen which has invaded our body earlier. So it is all about the antibody production. Hope all of you enjoy this video. Thank you.